In this lecture, we are going to discuss about SOLID, that is sequencing by oligonucleotide ligation and detection. So this is also a next generation DNA sequencing technology which is developed by Life Technologies, which is now known as your Thermo Fisher Scientific under the brand name of Applied Biosystems, and it has been available commercially since 2006. So this is a next generation technology which generates 10 raised to 8 to 10 raised to 9 small sequence reads at one time. Okay, and it is also one of the methods which is not using your sequencing by synthesis method, but here we are using sequencing by ligation. So it uses two base coding to decode the raw data generated by sequencing platform into sequence data. So let us go forward, let us understand how this works. So SOLID is an enzymatic method of sequencing that I said that it uses ligation, that means the enzyme DNA ligase. So we all know DNA ligase has been used widely in biotechnology, and it has the ability to ligate the double-stranded DNA strands. Okay, so emulsion PCR is used in this method to clonally amplify the templates. Okay, so you get many single-stranded primer binding regions. So these are the adapters, okay, so which has been conjugated to your target sequence. These adapters are linked to your target sequences, and then this entire your complex is then linked onto the beads. So these beads are then deposited onto glass surfaces. A high density of beads can achieve, which in turn increase the output of your technique or throughput of your technique. So let us have a review of the ligation chemistry process. So these are the steps. The primer first binds to the template strands, and then the probe hybridization. So once the probe and primer both have binded, the ligation takes place between them. The fluorescence is being measured after that. Once the fluorescence has been measured, the dye end, the fluorescent dye is being cleaved off, and these entire 1 to 4 steps are being repeated 6 times again, so total of 7 times. So this entire process then have to be completed 5 times. Each time primer is offset by base 1. So I will explain this entire process one by one in my next slides. So what happens is, once your bead deposition has taken place, then a primer of say length n goes and hybridization. So what happens is, this length n is going to be mostly equal to the length of your adapter. So then the beads are exposed to the library of 8 MER probes. Okay, that means there are 8 nucleotides or this probe is going to be made up of 8 nucleotides which have different fluorescent dyes at your 5' end and the 3 hydroxyl group at your 3' end. So bases 1 and 2 are actually complementary to the nucleotides to be sequenced, whilst your bases 3, 5 are degenerate bases. That means they can bind to any of the bases which are there. And your 6 and 7 are also universal bases, which are your inosine bases, which are linked fluorescent dye. Okay, so just let me show you the next. These are your, this is the probe anatomy. So you can see here the first 2 nucleotides are your the dinucleotide permutation. So there are total 16 possible combinations which can be there. So for example, you have CT, CA, CG. So these are the 4 combinations, and CC, 4 combinations of 1. Then you will have AA, AT, AG, AC. So similarly, there are total 16 dinucleotide permutation combinations which are going to be there. Okay, the next 3 are your universal bases. As I have already told you, they can bind to any of the 4 nucleotides. And then again you have these 3 inosine bases. Again they are universal, and they are linked to your fluorescent dye which is to be measured. Okay. So now out of this out of the 16, only one of the probe which is complementary to your dinucleotide which are exposed after the primer, so only that particular probe will come and hybridize to your target sequence. For example, if the next sequence is GT, so the probe which has your CA as the first two nucleotides. So that particular probe will come and bind. Once it has come and bind, the DNA ligase will join this probe and the primer. It will ligate it. Then there is this phosphothiazide linkage which is present between your base 5 and 6, which allows your fluorescent dye to be cleaved from the fragment using your silver ions. Okay, so that is how once your fluorescence is being already recorded, then it is to be removed. Okay, so this is how I will show you that this is your primer. So this is your primer, this is your adapter. So the primer of length n, which is equal to the length of your adapter, that will come and bind, and now these are the nucleotides which are exposed. So now the nucleotide which is, or the probe which is complementary to the first two nucleotides of your target sequence, that particular probe will come and bind, and the ligase enzyme is going to link your primer to your probe. It will ligate it. Once it has ligated, now the fluorescence which is released from your this probe, because of the laser that excites this fluorescent dye, that fluorescence is going to be measured and recorded. Okay, so here for example it would be recorded that there is an orange color fluorescence. Okay, so that recording is going to be stored by your computer. Once the fluorescence has already been recorded, as we have already discussed, there is going to be a cleavage of your, this is your 5th nucleotide and this is your 6th nucleotide. So between that your phosphothiazide bond is there, so that is going to be cleaved off. So this entire section is going to be now cleaved off. So you can see here, after the first round, you can see that you are going to get just an incomplete data. That means you have this 5 nucleotides bound to it, but you know that only this orange color has been recorded. Still you don't know, because this orange color is going to be common for 4 different types of your probe. So you really don't know whether it is AG or anything else. And the next 3 are universal nucleotides. So you don't know which are those. So there are different rounds of these sequencing or pattern that has to be followed up. So cleavage allows the fluorescence to be measured. Once that is being done, so it will also generate a 5' phosphate group, which will help in the next probe to come and bind and get ligated. So once your first round of sequencing is completed, so I'll just show you how the first round of sequencing will look like. So here you can see that this is your 5 nucleotide sequence which will come, and next again now you will see that the 6th and the 7th position is going to be vacant, or it is getting exposed. So the next nucleotide will come and bind, or next probe will come and bind. So there the green colored fluorescence is going to be detected. After that you will see a blue colored fluorescence detected. So here you can see every 5th base the fluorescence measurement is recorded. It is not that every base the fluorescence is recorded. Okay, so I'll explain you later that how these could be then deduced, or how the sequence can be then found out. Okay, so once this first round has been completed, so this whatever your extension product is there, that extension product is melted off, and then second round starts. Now when the second round starts, the primer length is reduced by 1 base. So now the length of the primer is n-1. So many rounds of sequencing has to be followed up later on. So once this n-1 is done, then it would be n-2, then it would be n-3. So let us then check how it will look like, how the sequencing will look like. So here you can see that when it is n, so these are the your sequences which are going to be detected, or say the fluorescence which is detected. Next, when it is your n-1, now n-1 ho jata hai, to what will happen is the part of your adapter also, the last nucleotide of your adapter also is now exposed. So this is the A. A is complementary to that last nucleotide of your adapter, and G is complementary to the first nucleotide of your template sequence. Okay, so kindly remember that. So then n-2, here you will see that the last two, or your first probe is going to be complementary to the last two nucleotides of your adapters. And adapter sequence is an artificial new oligonucleotide. So you already know what is the sequence of the adapter, and that will help us in deducing what is the sequence of your template. Okay, so let us now find out that how exactly the sequence is to be deduced, or the sequence is to be found out, because till now we have understood that only the color of the fluorescence of each of this probe has been recorded. So this is a unique method of finding out. So you can see here it is very clearly the base and the color defines what is the next base in the sequence. Okay, so here it is not, the color will not tell you that which is its base, but the color will tell you which is the next base. Okay, so that is the reason why we have to start with this N-2. With the last nucleotide of the adapter is exposed, and the probe which is complementary to that comes and binds. Okay, so here we already know that it is an A. So that means we know that the last nucleotide of your adapter is T. So you can here see T, and which is the corresponding probe that is coming. So which is the corresponding fluorescence that is recorded? Orange. So now see this matrix. Okay, so this is T and the corresponding color is orange. So that means which is your next base? C. Is it right? Yes, it is C. Now see C, which is the corresponding fluorescence? Red. So C के लिए red, and so what is the color, or what is the base that should be there? Next base? G. See here, G. G, which is the corresponding color? Green. So G, green. What should be the next base? T. T. Am I right? Yes. Again T, the corresponding color is green. So which is your next base? G. So that is how you can continue, and you can find out what is the sequence of your entire template DNA strand. Okay, so this is again a very good method when you want to check the sequences of an SNP, where even a change in a single nucleotide is very, very important. So here there is a repetition, or you can say that the checking of your data, checking of the sequence twice. So here, for example, I'll tell you. So here you have G. So you can see in the next thing also, this is a common way. This is a common G. So that is how this particular position is checked twice, whether it is G or not. Here you can see, here it is C, here also it has to be C. It is A, here also it has to be A. So twice every position is going to be checked, and that is the beauty of this particular method. So this is just an overview of the entire process. You can see the step 1. Here the primer comes and binds, which the primer is going to be of the same length as your adapter, and then there is the probe that will come and bind depending upon what is the next two nucleotides which is exposed. So here you can see TA is exposed, hence the probe with AT comes and binds. Next, the fluorescence dye is excited, and you can see here the fluorescence is measured. So once the fluorescence is measured, the fluorophore is going to be cleaved off, and this entire process is going to be repeated. So here you can see that every fifth nucleotide, the fluorescence is going to be recorded. Once this round is completed, then this extension product is melted away, and a new primer is going to come with a different length. Okay, so that is how your entire sequencing of DNA using your solid method can be accomplished. So two-base sequencing method, since each base is effectively sequenced twice, I have already explained you how. This solid technique is highly accurate. So at 99.99% with the sixth primer, it is the most accurate of the second generation platforms, and also comparatively inexpensive. So it can complete a single run in 7 days, and in that time can produce 30 GB of data. Unfortunately, its main disadvantage is the read length is very short, making it unsuitable for many application. Also, many a times it has been seen that solid method causes a lot of problem for palindromic sequences. So such palindromic sequences may not be sequenced properly using the solid technology. So this is all about the solid technique. I hope I have able to explain the technique. Thank you.